Anyway, I've yakety-yacked enough. As the people start to trickle in, I will open this session for all of you here. Thanks for joining us today at a morning edition, at least for most of the viewers, I think, of the EUVEN seminar series. And I'm very pleased to be joined today by Maria and by Irina. Irina is as far away as the east coast of Australia. So for her, it's a bit later, and that's the reason why she will give her talk first. She's a professor at the University of Queensland, and she is the director of the Center of Pain Research at the Institute of Molecular Bioscience at the university, and it's located in Brisbane. And she has worked on a whole range of toxins from a whole host of organisms. And today she's going to talk about something that until recently was unspeakable in polite circles: venomous plants. So Irina, if you want, the floor is yours, and I look forward to the talk.
Thank you very much. And apologies to those of you who have seen this presentation before, but you can never talk too much about venomous plants, I think, personally. Can you see those slides okay now?
Perfectly fine.
Okay, fantastic. Today I'll expand a little bit on the story of the stinging nettle toxins that we've worked on for quite some time now. And as it turns out, they were quite handy in identifying a new sodium channel interacting protein. So usually I start this talk off with telling the audience why venoms are fabulous. And I'm sure you've all seen similar slides that essentially just illustrate the great diversity of venoms, but I don't think I need to tell this audience that venoms are a fantastic source of toxins. What I did want to highlight here today is that they're in particular a fantastic source of toxins to interrogate the function of voltage-gated sodium channels. The reason for that is that venomous animals and plants can use their toxins for both defense and predation. And as it turns out, voltage-gated sodium channels have a key physiological function that's conserved right across phyla. So it doesn't matter whether a venomous animal is hunting insects or trying to deter large mammals like humans. Targeting voltage-gated sodium channels is a very effective strategy for both defense and predation. And that's because voltage-gated sodium channels underlie the upstroke of the action potential in all excitable cells. So, for example, if you have, and I might just get my laser pointer, if you have a sodium channel blocker, you might end up with flaccid paralysis, or if you have a sodium channel activator, you might end up with spastic paralysis or pain. And all of those are obviously very useful for defensive or predatory purposes. So hopefully after today, in polite company, you will be talking about venomous plants, but I'm sure you're all aware that many plants from the nettle family inject secretions for defense via stinging hairs. And if you're in Europe, you would probably be familiar with Urtica dioica or the common stinging nettle. Maybe, Ronald, in New Zealand, you've come across Urtica ferox or the onga onga. In Canada, for example, you've got the Portia species, the Portia canadensis. And in Australia, we have a family of stinging trees belonging to the dendronite family. And that, in particular, today I'll be talking about dendronite excelsa. So as everything venomous in Australia, we do everything bigger and better. And the stinging trees are no exception here. So Australia has some of the world's most notorious stinging plants, not only in terms of their size. So this is a picture of the giant Australian stinging tree. You can see that that's significantly larger than a European stinging nettle. They can grow up to 35 meters tall. And they're found right across the east coast of Australia. And importantly, they're renowned for causing extremely painful and also long-lasting stings. Some of the species in North Queensland in particular are so allegedly painful that the council has even erected warning signs so that unsuspecting tourists are aware that if they come in contact with some of these plants, they might need to seek medical attention. Like other stinging nettles, the leaves and stems of the Australian gympie tree are covered with these fluid-filled trichomes. So on the left-hand side here, you can see that these trichomes are sort of needle-like hairs that give the plant a bit of a fuzzy appearance. But on a closer look, you can see that they are really sort of like hypodermic needles filled with a fluid. And on the electron micrograph on the right-hand side, I'm hoping that you can appreciate, for example, in this trichome in the background here, it's actually the little bulbous structure at the very tip that breaks off when you touch the leaf, and then this essentially leaves a hypodermic needle that can penetrate the skin and deliver this bioactive fluid, which perhaps by definition you could call a venom. So what are the symptoms of a gimpy sting actually like? So just like the European stinging nettle, when you come in contact with the leaves, you get sort of an immediate pricking and stinging pain, itching and piloerection. And so this is a picture of my arm after an unfortunate gardening accident. I do have a few specimens as pot plants around the house, and they're not always the easiest to take care of. And what you can see here maybe is sort of the development of little goosebumps pretty soon after contact with the leaves. After a few minutes, you then get a rapidly developing wheal, and that's really a symptom of a histamine reaction. And it's well known that the stinging secretions contain histamine, serotonin, and acetylcholine. And then you get a more slowly developing axon reflex flare and axon reflex sweating. So this is really local vasodilation that occurs as a result of activation of peripheral nerve terminals that then release vasoactive peptides in the skin. And the really remarkable thing about these stings is actually sort of the pricking, burning, weird crawling, shooting and aching sensations that last for hours. Sort of the minimum I think that I've experienced is probably about 8 hours. And also you can have reoccurrence of these symptoms definitely for days. And in actual fact, I think the longest I've ever experienced sort of flare-ups was about 6 to 8 weeks after a sting. And so that's what got me interested in these stinging trees initially. To cut a long story short, we isolated a family of 36 amino acid peptides with 3 disulfide bonds from the venomous secretion or the venomous fluid in the trichomes. And just in terms of nomenclature, for peptides from the dendronite excelsa, from dendronite excelsa, we call these excelsa toxin, and mostly I'll be talking about excelsa toxin A today. And then, for example, peptides from dendronite morvoides, we called morvoidotoxin or motoxin. Now, the interesting thing about these peptides is that they had absolutely no similarity to known peptide sequences. And the most closely related are actually these albumin-derived plant peptides. So, for example, PA1B or leg insulin, but you can appreciate that they're actually not really that similar. What was interesting, though, was that the cysteine spacing is very similar to animal venom-derived inhibitory cysteine, not venom peptides. So after we identified the gimpytides in the dendronite excelsa trichomes, we thought we wanted to confirm that these really are the pain-causing principle. And so we generated the peptide synthetically and confirmed bioactivity in an in vivo assay. And so here what you can see is that a local injection of excelsa toxin, so that's in the blues and the greens, or motoxin in the orange, into the hind paw of a mouse causes local pain-like behavior. So that's paw lifting, licking and flinching. Is a very complicated assay, involves a student, a timer and a clicker. And what you can hopefully appreciate is that administration of these peptides into the hind paw causes pain-like behaviors. And also similar to what you see after human envenomation, so that the axon reflex flare, you can also see local vasodilation in the hind paw of a mouse. So that's reflected in the increase in the paw temperature in the ipsilateral or injected hind paw. But perhaps most compellingly, you can see that in the thermal image on the right here, where the injected left hind paw is clearly much warmer than the uninjected right hind paw. Because we were interested in pain and pain mechanisms, we then turned to dissociated dorsal root ganglion or DRG neurons, which are the cell bodies of sensory neurons. And here we found that exposure to excelsa toxin causes calcium influx. So what you can see here is a culture of associated sensory neurons, where each of the dots is a single cell loaded with a fluorescent calcium dye. And then the white arrows highlight cells that respond to application of excelsa toxin with an increase in intracellular calcium. And to summarize this graph for you very succinctly, what we observed was that only neurons responded to excelsa toxin, but you also get lots of non-neuronal cells in these cultures, and none of those actually responded to the toxins. And you can appreciate that in the graph on the right-hand side here, where non-neuronal cells that are not excitable by depolarization with potassium chloride simply don't respond to the toxin. So that was a little bit interesting, because recently we've been doing a lot of work on pore-forming toxins, so at least we knew that we didn't have that on our hands. In order to understand the pharmacology of this peptide a little bit better, we turned to patch clamp electrophysiology. And what we observed here was that excelsa toxin causes a persistent inward sodium current that's largely mediated by tetrodotoxin-sensitive sodium channels. And so for the non-electrophysiologists in the audience, what you can see on the left-hand side here in the black is the inward current carried by sodium ions in response to a voltage step depolarization. And hopefully you can appreciate that excelsa toxin in the green here almost completely abolishes the inactivation of the channel. So this causes prolonged sodium influx, which is consistent with the enhanced excitability that we see both in vivo and also in our cultured neurons. The two TDX-resistant isoforms, so NAV 1.8 and NAV 1.9, that I expressed in DRGs, on the other hand, did not respond particularly well to excelsa toxin. And so we were interested in understanding the sodium channel subtype selectivity in a little bit more detail, because mammals have 9 isoforms of the voltage-gated sodium channels, and some of them are actually interesting therapeutic targets. Now, one particularly interesting target is NAV 1.7, and that's because it's critical for excitability of pain-sensing neurons or nociceptors. And we confirmed that excelsa toxin indeed inhibits inactivation of NAV 1.7, both in human stem cell-derived sensory neurons. So maybe I'll just draw your eye to the summary graph on the right here, where you can see that application of excelsa toxin in the dark green here causes this persistent current that I just showed you in the DRG neurons. And this persistent current can be blocked by a selective NAV 1.7 inhibitor, which is PN3A, actually a spider venom-derived peptide, and a very, very handy tool compound. And similarly, we also used a human neuroblastoma cell line called TE671. And again, here you can see the excelsa toxin-induced persistent current, and that it's blocked by a NAV 1.7 inhibitor. So we thought we wanted to explore the subtype selectivity in a little bit more detail. And we turned to HEC293 cells expressing NAV 1.1 to NAV 1.8. Now, interestingly, we first tested NAV 1.7, and what you can hopefully appreciate is that excelsa toxin unexpectedly had absolutely no effect on inactivation of the channel. So we thought, oh, okay, well, something odd's going on. Maybe we missed something. Let's try some of these other isoforms that we know are expressed in DRG neurons. So we next tested NAV 1.6, and again, no effect of the toxin. NAV 1.1 is also expressed in sensory neurons. Similar story, no effect of the toxin. Starting to scrape the bottom off the barrel now. NAV 1.3 is expressed, but the function's not clear in DRGs. Didn't matter, the toxin didn't have an effect. And then we just became increasingly desperate, and we looked at isoforms that are really not expressed in DRG neurons. NAV 1.7 is a central nervous system isoform. The toxin had no effect, and so on and so forth. You get the picture. The skeletal muscle isoform, NAV 1.4, the cardiac muscle isoform, NAV 1.5. None of them responded to the toxin. We also looked at a couple of different neuronal cell lines, including DRG-derived cell lines, because we thought maybe these HEC cells lack cofactor, but the toxin also had no effect on ND723 or F11 cells, or also SHSR5Ys, which are all either DRG-derived or neuroblastoma cell lines. So we were a little bit stunned. And we thought, well, perhaps the explanation is that you need co-expression of a sodium channel beta subunit. There's 4 known beta subunits, beta 1 to beta 4, and the hex cell lines that we have express all of the channel isoforms together with the beta 1 subunit. So what we did was we turned to a Chinese hamster ovary cell line expressing NAV 1.7, and we co-expressed beta 1, beta 2, beta 3, and beta 4, and we then went to examine the toxin effect. And as you can see in the green, again, somewhat disappointingly and perhaps surprising, excelsa toxin had absolutely no effect on NAV 1.7 without beta subunits or with any of beta 1, beta 2, beta 3, and beta 4. So we thought, well, there's obviously something missing because clearly the effect is there in DRG neurons and also the TE671 cells. So we thought we'd turn to a lenti CRISPR screen, and this was done in collaboration with Greg Neely at Sydney University, where we took advantage of the excelsa toxin sensitivity of the TE671 cells to perform a genome scale lentivirus CRISPR knockdown screen. So just very briefly, the way this works is that you infect your TE671 cells with a CRISPR lentivirus library, and then you select your cells in which you've randomly knocked down all of the genes in the human genome with a toxin, and you compare guide RNA enrichment compared to a non-toxin treated control. Now, there was only one problem, and that's that normally this sort of screen is done based on cytotoxicity, but actually excelsa toxin is not cytotoxic to TE671 cells, even though it inhibits inactivation of voltage-gated sodium channels in the cell line. So on the left, you can see untreated cells. On the right, you can see excelsa toxin treated cells. It's quite clear that they're very happy. But we sneakily took advantage of a very old assay that's actually used to assess responses to, for example, Sigua toxin. And when you co-treat the TE cells with excelsa toxin, as well as very low concentrations of veratridine and EUVEN, which is their sodium channel activators and a sodium potassium exchange pump blocker, you can see that the cells are then not very happy, and you can use this to functionally select toxin resistant clones. And so that's exactly what we did. And in this graph, what you can see is the analysis of the guide RNAs that are enriched in toxin-resistant cells. So this is on the right-hand side of 0. You've got all of the guide RNAs that were enriched in toxin-resistant cells. And on the left-hand side, you've got guide RNAs that were enriched in toxin-sensitized cells. So in other words, if you knock down any of these genes, the toxin no longer kills TE671 cells. Now, we looked at this list, and it was quite promising at first glance, actually, mostly because one of the genes we picked up was SEN9A, which codes for NAF 1.7. So that really confirmed that we did have a sodium channel toxin on our hand. We also found, for example, NED4L, which is well known to affect ubiquitination of NAF 1.7. But other than that, it was actually quite interesting because there's really not much known about any of these other genes. And so we thought, well, the easiest way to confirm whether any of these genes actually confer a toxin sensitivity is to co-express them in our HEC NAF 1.7 cell line. And so that's exactly what we did. So initially we used a high throughput fluorescent membrane potential assay where we measure depolarization induced by excelsa toxin in control NAF 1.7 cells and NAF 1.7 cells in which we co-expressed the gene hits. And from this graph, it's quite clear that there was only one gene that seems to confer toxin sensitivity, and that's this one here, TMEM233. In electrophysiology assays, we confirmed a similar phenotype. So again, to recap that in HEC NaV1.7 cells, the excelsa alone doesn't really do anything to the persistent current. But then when you co-express TMEM233, what you can see is that the toxin, just like in the DRGs, causes this massive persistent current where the channel just does not close anymore. And so we were pretty confident that TMEM233 was the target that was present in the DRG neurons, but that was missing in our HEC cell line. And clearly it implies that this is the actual toxin target. We also did the reverse experiment where we knocked down TMEM233. So this was again in the TE671 cells. So on the left-hand side here, what you can see is the effect of excelsa again on the sodium current, where again you see this very profound sustained current. And then when you knock down TMEM233, you essentially almost completely lose that effect. So just like in the HEC NaV1.7 cells, you now no longer have a toxin effect. And so this really confirmed to us that TMEM233 is the target of excelsa. We also then, of course, wanted to confirm that this is also the case in DRG neurons. And luckily for us, John Wood and James Cox at UCL happened to have a TMEM233 knockout mouse. And we were lucky enough to be able to patch DRGs from these animals. And so on the left-hand side here, again, you can see the effect of toxin on wild-type DRG neurons, where again, you get this dramatic persistent current. And then in neurons from TMEM knockout mice, this effect is very significantly reduced. And this is just summarized in this graph in the middle here, where you can see the persistent current induced by excelsa in wild-type mice. And this is largely abolished in animals, in TMEM233 knockout animals. We also confirmed that the excelsa-induced pain behaviors are reduced in the TMEM knockout animals. And you can see that on the left-hand side here, where the pain responses of the wild type are significantly higher than those in the knockout animals. So overall, we were pretty confident that TMEM233 is targeted by excelsa. Now, if you ask yourself, what is TMEM233, then you would not be alone, because we also had not heard of TMEM233 previously. The reason for that is because there's actually not a whole lot known about it. What we do know is that TMEM233 is a member of the dyspanin family. The closest paralogs are TRARG1 and PRRT2, both of which are also expressed in DRGs. TMEM233 was initially predicted to comprise two transmembrane helices with an extracellular N and C terminus. However, both PRRT2 and TRARG adopt this single transmembrane topology with an extracellular C terminus, a membrane reentrant loop, and an intracellular C terminus. And this is also what AlphaFold predicts. I won't show the data today, but we have actually confirmed that experimentally as well. So it seems that TMEM233, just like TRARG1 and PRRT2, are this essentially one transmembrane proteins. Now, in order to assess whether excelsa binding requires only TMEM233, NaV1.7, or both, we used bioattenuated excelsa and assessed binding of toxin by flow cytometry. And so I always find that the scatter plots are actually easier to look at than the bar graphs. But what you can see here is the fluorescence signal to a biotin excelsa that was probed with a dialite 680 streptavidin. And in untransfected HEC cells without toxin, you obviously get no signal. Then in untransfected HEC cells with toxin, you get a little bit of background, but it's quite clear that you get very dramatic binding in HEC cells that are just expressing TMEM233, so that you can see that here with the number of signal positive cells. On the other hand, if you just have NaV1.7, there is not very much binding. And then if you have both NaV1.7 and TMEM233, again, you get very similar binding to the TMEM233 expressing cells. And so this really suggests that the toxin predominantly targets TMEM233. We also confirmed this by confocal microscopy, and so this is essentially the same experiment, but what you can see here is on the left, untransfected hex cells where toxin binding is visualized in green, and you can see that it's not visualized because there's not much there. And then in hex cells expressing TMEM233, you see clear toxin binding and clear signal. Now, we were interested to know whether the toxin binds to the extracellular side of TMEM233 or the intracellular side, because if you remember, I can't go back right now, TMEM233 does really not have much of an extracellular domain at all. It's essentially just 3 amino acids. And so the experiment we did to determine that was to include excelsa toxin either in the extracellular solution, which is in the green, or in the intracellular pouch solution. So that's in the red. And again, what you can see is that the toxin has no effect in the absence of TMEM233. This is in cells expressing NaV1.7. As I've shown you previously, if you co-express TMEM233 with NaV1.7 and have toxin in the extracellular solution, you get a very profound persistent current, but toxin in the intracellular solution has no effect. So my interpretation is that the toxin likely accesses TMEM233 and NaV1.7 through the extracellular side. So in summary, I've skipped over some data that I didn't show today, but you can either ask me questions or you have to believe me. But hopefully I've shown you that excelsa toxin is a pain-causing plant venom peptide that inhibits inactivation of tetrodotoxin-sensitive voltage-gated sodium channels, in particular NaV1.7 in DRG neurons. The effect of excelsa toxin occurs via TMEM rather than a direct interaction with the NaV1.7 alpha subunit, which is quite unusual, and the first case of any toxin that I know that modulates sodium channels in that manner. TMEM233 is a really intriguing, quite small protein, a member of the dyspanin family that can associate with NaV1.7. And so we propose that TMEM233 is a novel sodium channel interacting protein, and that co-expression of TMEM233 both confers excelsa toxin sensitivity and also subtly modulates the gating parameters of NaV1.7. This was actually a lot of work involving a lot of people, but I just wanted to highlight in particular some people from my group. Sina Jami, whose main PhD project was on this, Jennifer Doosan to be our class closer, as well as Sam Robinson, that were well known in the toxin world, hopefully. From the Uni Sydney, Greg Neely in particular, for his help with the CRISPR screen. Steve Waxman and Sulaiman Dibhaj from Yale, who helped us a lot with some other work that I didn't show today on DRGs. And then also at UCL, James Cox and John Wood, especially for the TMEM knockout mice. And I will leave it by saying that the EUVEN is a fantastic initiative, and I think Australia should be like Eurovision, where we should become honorary members. So
thank you, Irina. That was actually the most interesting plant talk for me personally that I've seen in a while, because it's a great detective story. I mean, this is way outside what I can claim even the most marginal expertise in, but it sounds like a long path that starts in exasperation, but you keep going, you keep going, you keep going, and then there's a payoff. So very interesting. It's most questions I was jotting down. Well, let's not talk. Let's ask the audience if there's any questions or comments. The floor is yours, and if you're too shy, you can type them in the comments.
I'm also available offline.
Also
online.
You can, yes, you can always send her an email. So I was thinking more about what does the plant do with it? Do you think toxins like this evolved specifically to deter large herbivores like mammalian leaf eaters?
I think that's the general consensus at the moment. we have tested the insecticidal activity of the peptide, and it does absolutely nothing. Maybe not surprising, but so it doesn't seem to be insecticidal. What exactly, what herbivores are supposed to be deterred is not really clear. There's also stories, for example, of some Australian, the paddy melon eating the leaves and being resistant to the toxin effect.
What's a paddy melon?
It's like a small sort of forest kangaroo.
Okay.
Yeah, I'm not really sure.
Well, there must be something, and it must cost the plant an enormous amount of metabolic resources to produce these structures and these toxins.
Definitely. I think the general consensus is that they're defensive toxins, but against what? And we will also, I mean, some of the work that we're doing now is to look at the defensive plant toxins more broadly across different species, and also to try and reconstruct, how widely they occur. At the moment, they seem to be unique to Australian stinging nettles. We've done some other work that I didn't show today on the European stinging nettle and the onga, and they also have sodium channel toxins, but they're more like spider or scorpion venom peptides, where they just conventionally target the alpha subunit. So these seem a little bit weird.
How did you collect the venom? Because the structures that actually they are microscopically small, did you shave these off, or how do you?
So Tom Durek did that in. They've got a lot of expertise with plants. Actually, that he basically just homogenized it in with liquid nitrogen. You just sort of break it off. There's another story to this, which this started a long time ago, sort of as a side hobby. The kind of project that you would never ever get funding for, and you just do on the weekends. Where I spent there's been reports from the 50s and 60s that there's a cyclic octapeptide called muroidin that causes the envenomation symptoms. So I spent a couple of years trying to find muroidin in Dendronite excelsa, and I could never find it. The reason is that it's not there. It's only in Dendronite muroides, and also it doesn't actually do anything. So that's another story. But yeah, so we've tried all sorts of ways of extracting the toxin and compounds. So yeah, homogenization, extracting, just shaving the needles off, and all sorts of things.
Well, that would have been a shorter talk with a far less satisfying end.
Yeah, definitely. It's not there and it doesn't do anything.
Anybody else? Any question?
Can I ask?
Sure.
So do you think TMM interacts physically with the sodium channel, or it starts some kind of signaling cascade which eventually affects the sodium channel?
That is the $1 million question. So I'm hoping it's a direct interaction, but we haven't got particularly good evidence. The boring molecular biology and patch clamp data that I didn't show today was we've truncated various bits of TMM, and when you truncate the N-terminus, you lose the TMM sodium channel interaction. But on the reverse, if you synthesize the N-terminus of TMM and include it in the patch pipette, you can actually see similar effects. That's the most evidence I have at the moment, is that there might be a direct interaction. But other than that, we haven't been able to show it with anything yet. But I suspect it's a direct interaction.
Okay.
I'm hoping it is.
Okay.
Well, thank you, Irina. There's a question in the chat, but I will leave it to you to answer it in typing by Harriet. It's a good question. Thanks very much for coming to joining us in the evening from where you are. It was a very enjoyable talk. I hope to at some point see you again in person. And we move on to our next speaker, Maria Sachkova. She is currently a lecturer in the School of Biological Sciences at the University of Bristol, just west from here. And she, for me, is famous for having done some very outstanding work on the venom system of Nematostella vectensis, the model sea anemone. But she's also worked on spider venom toxins and on neuropeptides and the nervous system structure in comb jelly. So a broad tableau of interests. And today she's going to talk about her work in the Matostella. So Maria, thank you for joining us, and the floor is yours.
Thank you very much for the nice introduction and invitation to this nice seminar. So yes, today I will be talking about ecology and evolution of venom in Cyanemone nematostella. And even though currently I am in the University of Bristol, this work was done when I was a postdoc with EUVEN Moran in Israel, which is right here in the middle of the globe. And I used to work on the ecology and evolution of the venom in Cyanemone nematostella. So this is a really great venomous organism, because unlike many other organisms, it's a quite well-established lab model. So you can culture these animals in the lab, and they will be available throughout the year. And you can do a lot of advanced molecular biology methods in it. For example, you can make transgenic animals, and this will allow you monitoring the toxin production, the dynamics of toxin production in different conditions, and you can do it in very great details. So let's talk about the ecology of this Cyanemone nematostella. So as all the cnidarians, it is a venomous organism, and it also goes through the complex life cycle. When nematostella is adult, it looks like a polyp, which sits in the mud in the bottom of these lagoons, and it only leaves the tentacles exposed outside. And when it is young, it is represented by a very small motile planula or larva called planula. And there is a difference between the adult and the larva in terms that the larva doesn't feed, and the polyp does feed. So because of the differences in the size, in the feeding behaviors, in the lifestyle, the interactions with their natural predators in prey differ throughout life cycle of this animal. And examples of natural predators are grass shrimps and some fish called mummichog, and natural prey can be insect larvae, which live in the same lagoons. Yes, so here are some examples of nematostella hunting behavior. So when you try to feed it with the small Artemia shrimps, they get stuck to the tentacles, and then they get killed by the venom, and nematostella puts them into the mouth. So as you probably know, the venom is contained within the stinging cells called nematocysts. And when you touch the animal, they get discharged and they inject the venom. However, part of nematocysts, there are other kinds of venom-producing cells called ectodermal gland cells. So they just secrete a lot of venom components. And both cell types are distributed throughout the body of the adult polyp and the larva planula. Basically, both life stages are venomous. And also, unlike many other venomous organisms, these animals don't have any defined venom gland. The stinging cells are distributed. The stinging cells and the ectodermal gland cells are distributed throughout the body. And now I will be talking about several of my projects, which I've done during my time as a postdoc. So the first one is about the mechanism of toxin evolution in this Cyanemone. So nematostella has multiple toxins, and one of the major venom components is called NV1. And NV1 is produced by ectodermal gland cells, and it is known that it is highly toxic to shrimps. So if you inject NV1 into shrimps, they will die immediately. And if you incubate fish larvae with NV1 toxin, it will also affect them, but a bit later, within 1 hour. And then there are several homologs of this toxin. 2 of them are called NV5 and NV4, and they have quite different activity from NV1. So both of them don't have any effects on the shrimps, but they do affect fish. So fish will be killed, like here we use fish larvae, they will be killed within 30 or 40 minutes. Another difference between this NV5 and NV4 and NV1 is that these 2 toxins, NV4 and NV5, are larval toxins. They are produced in the ectodermal gland cells of larvae, while NV1 is an adult venom component. So if we look in the evolution of this venom toxin gene family, we can assume, or we can propose a mechanism of the evolution of these genes. So most probably there used to be an NV1-like ancestral gene, which probably was very similar to NV1 with the anti-shrimp activity and strong sodium channel inhibiting activity. And then there were multiple gene duplications, sometimes which could have been either in the same genome locus, or they could have been accompanied by translocation to a distant genomic loci. And then different copies underwent different processes. Multiple NV1 copies were highly conserved through the through the consorted evolution, and this has been shown by you quite a long time ago. So NV1 has multiple copies. It's produced in very high levels, and all the copies are nearly identical. And all these products have strong anti-shrimp activity and weak anti-fish activity. And then NV4 and NV5, their sequence diverged quite significantly from NV1, and they acquired new functions. So they stopped being an adult toxin, they became a larval toxin. And on the other hand, they acquired a strong anti-fish activity and lost anti-shrimp activity. And this is one option, or if the ancestral gene combined all of this function, then what happened to NV4 and NV5 was that they underwent subfunctionalization, where they retained the only part of the activities, basically anti-fish activity, while NV1 retained the anti-shrimp activity. Also, there are several pseudogenes which just have no function. So we can say that these toxins of this big gene family evolve through the birth and death mechanism in nematostele. Okay, so in the next project, I studied a different toxin family, and it is also about the evolution of new components in this family. So we know that the stinging cells on nematocytes, they are related to neurons in different ways. So first of all, both nematocytes and neurons share the same precursor in the development. So they develop from the same precursor cell on one hand. On the other hand, if we look at the structure of these stinging cells, we can notice that first of all they normally have a sensory structure on top, like an endocell. So if you touch it, the signal gets detected. And on the other hand, in the bottom, they have these long processes which look pretty much like neurites. Also, nematocytes, they are physically connected to neurons through synapses. So there is probably a neural regulation of nematocyte activity. And this is how it looks if you touch sea anemone and they discharge the venom. Yes, I wanted to mention that unlike neurons, nematocytes evolved this capsule which has venom. So that's an innovation of this cell type. Okay, so in this project, we wanted to find new components of the venom, which would be similar to the well-characterized potassium channel toxin from Cyanemone stichodactyla. So that toxin is called SHK, and we searched for new components based on the similarity, on the sequence similarity in the nematostellar transcriptome. And we found several new sequences which look like a putative venom precursor, toxin precursors. So they had this part with several cysteines, which were arranged into the same pattern as in the SHK toxin, and then there is a cleavage site here. So most probably this is how the precursor gets cleaved, and the mature peptides, basically shorter peptides with several 3-disulfide bonds. However, surprisingly, it turned out that only one of this gene was produced by stinging cells. So if we look at this in situ hybridization staining, we can see that in both planula and polyps, this gene is expressed in the cells which have this empty spot in the middle. So this empty spot corresponds to this capsule, and the staining is RNA based. So RNA is all around this capsule. So that's how we can recognize the cells as nematocytes. And then if we purify the capsules, the nematocysts, and run proteomics, we can detect this SHK-like-1 peptide among other known venom components of nematostellum. So this suggests that this gene SHK-like-1 is indeed produced by stinging cells and is probably a toxin. However, other new peptides were localized to neurons. So you can see here the cells with long branching neurites, and they correspond to ganglion cells of the nervous system in nematostella. Other homologs were localized also to other cell types like sensory neurons, but I'm not showing here. Okay, so two of these new genes, SHK-like-1 and SHK-like-2, have quite similar sequence. They're quite similar in their sequence and also in predicted structures. However, one of them is produced by neurons, SHK-like-2, and another one is produced by stinging cells. So we wanted to characterize the functional activity, and for that I produced recombinant peptides and tested them on the zebrafish larvae and also on the polyps itself. So both peptides turned out to be toxic to zebrafish larvae with a small difference. It looks like SHK-like-1 is a bit more potent against the zebrafish larvae. And if we incubate nematostellar polyps with these peptides, surprisingly, both of them provoke contraction in these polyps. So maybe this reaction is not that surprising for SHK-like-2 because it is produced by neurons and probably it has some internal regulatory function. So it probably acts as a neuropeptide. However, SHK-like-1 is produced by the nematocyte, so it's supposed to be venomous venom component, but it still retains this function, this ability to affect the nematostellar body itself. So if we take a look at the phylogeny of these genes in Cyanemones, we can see that last common ancestor of all the Cyanemones probably had a gene, SHK-like-2 gene, which was probably neuropeptide produced by neurons. And SHK-like-1 is detected only in nematostella. So this SHK-like-1 production by nematocytes is a nematostella-specific feature. And this let us propose a scenario where probably there used to be an SHK-like-2 neuropeptide gene, which underwent duplication in nematostella. And then one of the copies retained the neuropeptide function, while the other copy moved into the switched expression into the venom system. However, because they have they share the evolutionary origin and they still share a lot of sequence similarity, their function in vitro test looks quite similar. Both of them still retain activity against fish, and they can also provoke contractions in the nematostella. But because they're produced by different systems in vivo, it's likely that the neuropeptide function is only typical for SHK-like-2, which is produced by neurons, while the toxic function is probably typical for SHK-like-1, which is produced by nematocytes. But anyway, this mechanism is an interesting example of recruitment of components of nervous system into the venom system. And now I want to quickly show you two projects which are showing that venom of nematostella is regulated based on the interactions with multiple biotic and abiotic factors. So this is the more ecological part of the talk. So first of all, we showed that that venom production is metabolically expensive in nematostella. So if we treat nematostella and make it discharge its venom, and after that we measure the consumption of oxygen, we can see that oxygen consumption increases after the discharge. So it means that nematostella consumes more oxygen and its metabolic rate increases. So to replenish the venom, it needs to invest a lot of energy. And using transcriptomic, we also show, sorry, it was encounter measurements. We also showed that after the discharge, the toxin production increases. So indeed, once you discharge all the venom components, they have to be remade, and that costs a lot of energy for nematostella. So because it's so metabolically expensive, the venom production is probably regulated in nematostella. So we checked how the venom we checked the dynamics of venom production throughout the different life stages. And as you can see here, some toxins are highly enriched in the larvae, some are highly enriched in polyps, and some are even enriched in the very early developmental stages like eggs. So here is an example for 3 of them. You can see that this toxin, for example, is absent in eggs, and then in larvae, it starts being produced, and then it retains the same level throughout the life. On the other hand, there are larval or egg-specific toxins, which are highly expressed in the beginning of the life cycle, and then in the polyps, the expression drops, and then peaks again in the females, which produce eggs. So this is an example showing that an example of a transgenic animal where the toxin was labeled with fluorescent protein, and we can see that it is expressed starting from the larva and then through metamorphosis, and it is also present in the polyp. And this transgenic approach allows to see that this peptide is localized to nematocytes, and also that not every nematocyte expresses this toxin. So we can see that there is a difference between toxin producing cells. Okay, so these differences in venom production suggest that it is maybe due to different interactions with different prey and predators. So here we did an experiment where we placed a nematostella into its natural or almost natural environment, where basically it was sitting in the mud. And here you can see a shrimp coming. So this shrimp, it quickly jumps away once it comes into a contact with nematostella. And this actually correlates with the fact that that adult nematostella produces a lot of anti-shrimp NV1 toxin. On the other hand, if we incubate nematostella larvae with a fish larva, fish will try to eat the nematostella larva. However, obviously it doesn't really like it. It spits it out, and that correlates with the fact that the nematostella larva has multiple toxins which are efficient or active against the fish. So because there is a high metabolic cost involved into venom production, it's very probable that there is a regulation in venom production, and for example, larva doesn't produce much of NV1 toxin, but instead it is focused on production of the toxins which deter fish. So this is important for the survival of the animal. On the other hand, the adult polyps are focused on production of the anti-shrimp toxins, because shrimps can eat the polyps basically. And in this project, we checked how the toxin production is regulated according to the abiotic factors. So we sampled multiple populations of nematostella, and like starting from the north to the south in the North America, and then and this population live in quite different climatic conditions, and then we subjected them to the heat stress and checked the expression of changing the expression of heat shock proteins and NV1 toxin. NV1 is really highly expressed toxin, so its production was significantly reduced in multiple populations in the north. However, in the south, in North Carolina population, the production of NV1 toxin was not really affected. So to understand it in more detail, we focused on this northern Massachusetts population and southern North Carolina populations. And at the next step, we basically checked more the expression of more venom components. So yes, basically the North Carolina population, it lives in much warmer weather conditions. So this is an experimentally measured temperature in the water during the 1 year, during 2016. And this is the heat shock that we used. So we can see that there are many days in the south where the temperature naturally reaches this 36 degrees, while in the north, there are way less days when the temperature reaches 36 degrees. So it seems that southern population is adapted to this hot weather, and because of that, when we subject it to the heat stress, the toxin production in the south doesn't really change, while in the north, it reduces a lot. And in the nature, the stresses don't come one by one. So normally the heat stress would be combined also by increasing the salinity and by high UV light exposure. So we also checked out this combined stress conditions, and we saw that there were a lot of effects on gene expression, but importantly, NV1 toxin gene was the most downregulated gene in the northern Massachusetts population. And this trend was also detected at the level of protein. So we did mass spectrometry, and we saw that in Massachusetts, the production of NV1 is less after the stress compared to the North Carolina population. So now let's conclude all this work. So basically, during my postdoc time, we managed to show that toxins evolve through the birth and death mechanisms, and also through recruitment from nervous system to venom in the Cynaemone nematostella. And then we could also show that venom composition is regulated across the life cycle according to the interaction with predators and prey. And because the venom has high metabolic cost, the environmental stress regulates the venom production, and the reactions to the stress differ between populations adapted to different environmental conditions. And now I want to briefly mention what I am planning to do, since now I moved to the University of Bristol and started my independent lecturer position. So hopefully I will be able to go back to study venom at some point. And so far I was focused on the non-symbiotic sea anemone Nematostella. However, many other sea anemones and their relatives, corals, they are symbiotic animals. And it's known that symbiotic corals, they form very rich ecosystems, like coral reefs. However, when you stress corals, they bleach, and basically they lose their symbionts. And this is a big stress for this animal, because symbionts provide sugars to the animal, to the coral. So if the coral spends too long time without the symbionts, eventually it dies. However, it's also known that both sea anemones and corals, they can prey, and they prey on this, for example, small invertebrates, and for that they use venom. And it's also known that when a coral is bleached, the importance of heterotrophic carbon increases relatively to the symbiotic state of this coral. So these facts suggest that feeding on feeding and preying might be a key factor allowing corals to survive bleaching. And venom is basically a key component of the predation behavior in these animals. However, even though it's known that corals have nematocytes and some toxins have been isolated, it's completely unknown how the venom is involved in the role of the venom is in bleaching is completely understudied. So I'm thinking of studying venom as an evolutionary adaptation to overcome short bleaching stress in corals. And for that, I will compare bleached and symbiotic animals, and we'll do it through different populations living in different environmental conditions, and also across different life stages, using molecular methods such as transcriptomics, transgenics, and also toxicity tests on the native prey. And at this point, I would like to finish and thank all the collaborators and former colleagues who were involved in these projects, and many thanks for listening to me. So yes, I'm done.
Thank you, Maria. I will clap with my actual hands. That was excellent.
Thanks.
I was already aware of a big chunk of your previous work, but your new research sounds really exciting. Before I make a comment on that, is there anybody who has a question or a comment?
Maybe.
Yes, Gregor.
Hi.
Hi.
Hi, Maria. Hi. A very, very nice lecture. Fantastic results. Very nice set of different methods that you use, and it's really nice to see how far you took this project and topic. Maybe just two brief questions. I found it interesting that you find RNA in nematocysts. Did I understand correctly?
Not exactly. So nematocyst sits within the cell, nematocyte. So RNA is outside of the nematocyst. So RNA is the cytoplasm of nematocyte. So that's why when you look at the staining, you see like an empty part, which is nematocyst. It doesn't have RNA, and RNA is all outside. Right?
So in your research, you were studying these smaller peptidic toxins detect on ion channels, but in Nematostella there are also performing toxins.
Yes,
No, is it true?
I think there are. Okay, right now, to be honest, I don't remember exactly, but it's most probable that there are some. But I was focused on this, on the cysteine rich.
No.
Sorry.
The question is whether it is known what's the biological role of these other toxins. Do you have any idea where they could act, or why would Nematostella produce them?
Right now, to be honest, I don't remember many. There are some. Yes, but sorry, I don't really remember.
It's off topic, yes. Thank you very much. Thank you very much.
Thanks, no problem.
I see that in the chat, Irina has posted a question. Does NV1 have effects on humans, and what could be the target?
I think NV1 has an effect on sodium channels of in arthropods. So I think it's quite selective for the arthropods. Like even though it has effect on fish, it's much slower. So like fish is a close relative to humans relative to arthropods. So I guess it probably may have some, but much weaker. Like if you touch Nematostella, nothing happens to you. So yeah, but if you inject NV1 into shrimps, they die right away. So it's very strong for the for the arthropods.
Any other comments or questions? I'm just very excited about your new project as well, because it touches on an incredibly neglected question, at least in the phenomics community, which is venom plasticity. It's like whether with a given genotype you can create, depending on environmental triggers, different venom compositions or expression levels. So that's why the work on environmental stressors that you did with Jay, who was already cool, but what you're going to do now with the bleaching, I think that's an excellent and original thing. I look forward to it. I mean, I'm traveling to Madrid tomorrow to get some sea urchin tissue samples sequenced, because I'm sort of interested in a similar question, at least conceptually similar. As environmental change and climate change happens, we want to use that as a way to give funders an opportunity to give us money. It's like, look, climate change can affect venom systems, perhaps, if they're plastic, if you change pH and temperature and salinity and all that stuff, if that messes around with their defensive and predatory weapons, we should study this. So I've done an experiment in Scotland where we basically exposed sea urchins, predator triggers, environmental predator triggers or not, to see if they will change the expression of defensive toxins, because that's never been done. And they are a very good system for studying plasticity in any way. So tomorrow I'm going to go bring those to the sequencing lab and see if we can get some data out. So I look forward to that. But yeah, I think that whole topic, anybody here study venom plasticity, we need to know more about this, because in the best studied venomous taxa, take snakes as the paragon, there's not much plasticity. I'm not sure if a lot of work has focused on studying the topic, but if you take a Bothrops from the jungle, you keep it in the lab for 20 years, feeding it dead mice, its venom profile is stagnant. So maybe, so it's these invertebrates we love so much we should study.
Yes. Yeah, hopefully. I'm really excited about this topic. Hope I will manage to do something in it, but yeah, I think it would be cool. It would be cool to just like, this coral bleaching is such a big problem, but nobody is really looking on the natural adaptation, or maybe people do look, but not on venom as a natural adaptation, which may help corals to, like, because for short time they can survive without their symbiotes. It's just that when it's long, they die. So maybe if we know more, it may be useful in some way. Yeah.
Okay, if there's no further comments, Maria and Irina, again, thank you so much for joining us today, for talking about your interesting work. I hope we all of us we'll be able to see each other in person again. I'm sure that somewhere in the first or second week of April, we will have another edition of this seminar series. So I hope to see you back then. For the Irina, sleep well later on. It's late for you. And for the rest of you, I hope you have a productive day.
Thank you. Bye.
Bye-bye.